Budding. It is a form of asexual reproduction in which an outgrowth develops on the parent, and this later becomes detached and is then an independent organism. So you can have, this is a parent. From this parent, an outgrowth develops. As time goes by, this outgrowth becomes detached, and then it becomes a fully independent organism. Budding occurs in organisms such as flatworms, hydra, and fungi such as yeast. So we look at budding in yeast cells. As an example. So here is a diagram of a yeast cell with the normal cellular inclusions, the cytoplasm, the nucleus. There is a subvacuole and food reserves. So yeast is a single-celled fungus that exists in many different species. Budding begins with a small area of the cell wall softening. So a particular area is going to soften on the cell wall. Now at the same time, while this is happening, a bud-like outgrowth develops on the parent cell. So this bud-like will develop on the parent cell. And while this is taking place, the nucleus here divides into two. Then one nucleus moves into the bud, and the other one remains in the parent cell. The bud then either separates from the parent cell or may remain attached. So once the nucleus has moved into the bud, a wall develops to separate the two. Now the daughter cell may detach from the parent or it may remain attached. If it remains attached, you can have another bud developing on the daughter cell and even another bud developing on the parent cell, such that when conditions are favorable, you can have many buds forming, thus making a chain of yeasts that are still attached to one another, especially when external conditions are favorable. That is, before a daughter cell has detached, it develops another bud, and the parent cells still continue forming new buds. And that's how this chain of yeast cells get to be formed. Now here is a photograph of budding in yeast cells as seen under the microscope. You can see the individual cells and how budding occurs in these individual cells. Here is one long continuous chain of budding yeast cells.